Welcome everyone to Ecology Live. I'm Pippa Gillingham, I'm chair of the BES Climate Change Ecology Special Interest Group, and I'll be your chair for Ecology Live today. If this is your first time joining us, you're very welcome, and welcome back to all Ecology Live regulars. The British Ecological Society started this series of 30-minute talks last year in the midst of coronavirus lockdowns. We're very pleased to say that we've got more great speakers lined up for another 7 weeks. You can check who is set to speak on our website. While you're there, why not sign up to receive our newsletter or find out more about our 7 world-class journals? Before we start, I'd like to say thanks to our sponsors. We'd like to thank Wildlife Acoustics for the wonderful moorland soundscape that started this session. We would also like to thank Oxford University Press, who are sponsoring this whole season of 12 talks. You can find details of their Ecology Live reading list, which includes discounts and some free content, on the slide at the end of the talk. This talk is being recorded and will be added to the BES YouTube channel afterwards. You can submit questions via the Q&A box at the bottom of your screen throughout the talk. There's no need to wait until the end. There will be a short Q&A at the end of the talk where I will read out questions to Lauren and she'll be able to answer them. Your questions can be named or anonymous, you decide. You can also upvote any questions you find particularly interesting by clicking the thumbs up icon next to the question in the Q&A. And now, it gives me great pleasure to introduce today's speaker, Professor Lauren Buckley. Lauren is a professor of biology at the University of Washington. She achieved her PhD from Stanford University in 2005, looking at community ecology and biogeography of lizards on islands. Since then, she has moved on to understanding how biology, including morphology, physiology, and life history, determines an organism's ecological and evolutionary responses to environmental change. She's worked on a wide range of taxonomic groups, including more recently montane butterflies and grasshoppers. Along the way, she's held postdoctoral fellowships at the National Centre for Ecological Analysis and Synthesis and the Santa Fe Institute. She's been recognised as an NSF Career Awardee, a National Academy of Sciences Cavalry Frontiers of Science Fellow, and a Future Leader at the Science and Technology in Society Forum. Today, she's going to talk to us about forecasting ecological and evolutionary responses by characterising how organisms experience climate change, including some really exciting sounding tools to help us understand how organisms experience their environment. Over to you, Lauren.
Great. Thank you so much, Pippa, for the introduction. And it's really nice to be here and have the chance to interact with all of you. See if we can get this sharing started.
I can see your screen, but it's not quite centered on the slides. There we go.
Great. Okay, yeah, so today I'm going to go through a case study of why I really think we need to think more about how the biology of how organisms are interacting with their environment. And then I'll be going through some tools to be able to better relate the environmental conditions to organismal responses. So to me, a really foundational and interesting challenge is being able to forecast species responses to climate change. And what really motivates my research group is the observation that species are responding quite individualistically to climate change. So here we have an example from the Grinnell Resurvey project out of the Museum of Vertebrate Zoology at the University of California, Berkeley, where they've gone back to Yosemite National Park and other areas following about 100 years of climate change. And in this resurvey project, and in general, we do find that organisms are moving uphill and they are moving to higher latitude as we expect. But there is also a lot of heterogeneity, as you can see here, across these different taxa. So as biologists, we know a lot about species traits and how those species traits mediate their responses to the environment. But when we do these fairly coarse modeling approaches trying to relate traits to the magnitude of climate change response, we're really not able to do a very good job right now. So you can see here a slide from a synthetic study where for many taxa and many traits, there was no significant relationship between the trait and the magnitude of range shifts. And of course, there's reasons for this. Usually we don't account for things like thresholds and nonlinearities and interacting traits. And we really need to think more about the biology by which these traits are mediating responses to climate change. And so the International Intergovernmental Panel on Climate Change has recently been focusing on questions such as, what does a 1.5 or 2 degrees Celsius temperature increase mean for organisms, ecosystems, and biodiversity? So in the context of climate change initiatives such as the Paris Climate Agreement, we really need to know what a given amount of temperature change means for different types of organisms and ecosystems. And we have some ideas, as you can see in this graphic here. We can make generalities about the magnitude of change in the response. But in my perspective, we really don't have the tools we need to appropriately consider how organisms respond to climate change and make the detailed predictions we need going forward. So what are some things we need to consider more to improve our predictions? I would argue one of the main things we need to do is to think beyond air temperature to understand how organisms experience climate change. So here we have a paired visual thermal image of a sea star on a bed of mussels in the intertidal zone. And the most common way to forecast climate change responses would be just to use air temperature well above this intertidal landscape. But by looking at this picture, you can see that we really would have a challenge getting the right answer doing that. If we look at this thermal image, the two organisms, despite being in the same air temperature, are experiencing quite different temperatures. The mussel is a lot warmer than the sea star. And because sea stars are eating mussels, that's also going to have implications for their interactions. And they have different thermal and different physical properties that are affecting how quickly they heat up. So we can also see that continuing with this mussel example here, we're using data from Brian Helmick with robomussels, which are basically physical mimics to understand the body temperature mussels are experiencing in their environment. So again, if you look at this paired visual thermal image, you see a lot of heterogeneity at very small scales. And that heterogeneity can be very important for how exposed organisms are to the environment and to climate change. And if you look at those graphs, that's looking at maximum daily temperature through the year at different locations. And it's by intention, a very messy plot showing you a lot of lines just to highlight really how heterogeneous the experience of mussels are. And their thermal experience depends on where they are, what zone they are in the intertidal zone. So those are the different colors. And also, even within the zones, the different lines show that there's a lot of variation that these individual robomussels are experiencing. So we really need to think about both how exposed organisms are to the environment and then how organismally or physiologically sensitive these mussels are to the environment. Another real driving complexity in understanding responses to climate change is the occurrence in responses. So we have here a plot of phenotype as a function of the environment. You can imagine we start with that blue star there. So that would be an organism well matched to its environment through evolutionary history. We can then imagine climate change imposing a shift along the environmental axis, such that now the phenotype of the organism is not very well matched to what would be its optimal phenotype, which we're depicting with a red star now. So what's most often studied and what often is the case in nature is this tracking response shown in purple. So the organism might be able to track its niche through space or through time to return back to an environment where its phenotype is a good match. But that's often not possible in nature. So then we get into the other arrows where if an organism is going to persist, it likely needs to exhibit some combination of plasticity and evolution. So plasticity would be a shift in the phenotype in response to the shift in the environment. That might get an organism closer to its optimal phenotype, but there's still likely to be some gap there of phenotypic mismatch that evolution may need to play a role in improving the match between phenotype and the environment. And in particular, this interplay between plasticity and evolution, I think, is really interesting in the context of climate change. Plasticity can both facilitate evolution by allowing organisms to persist and buying them enough time to evolve, but it can also buffer selection and slow down evolution. So the interplay of plasticity and evolution is a major recent theme of our research that I'm going to talk more about today. And I want to go over the primary tool we use to really match up organisms in their response to the environment. We call mechanistic niche models. So you can start with large-scale climate information. We then use microclimate models to scale to the conditions organisms are actually experiencing in their habitat. We then can match that up with phenotypic information in these energy and budget models. So we are budgeting energy exchange between organisms and the environment to predict their body temperature. And again, as you can see in this example for a grasshopper here, the body temperature of a grasshopper or any organism can be quite different than their environment. So it's important to use body temperature often in these models. And I'll say these microclimate and biophysical modeling tools are some of the tools that I'll get to talk more about at the end of the talk. So once we have body temperature, we can then match that up with lab estimates of performance as a function of body temperature to predict organismal performance in a given environment. And we are looking at performances that have strong effects on survival and fecundity. So once we can estimate survival and fecundity consequences of performance, that gets us the fitness, and we can also project that onto landscapes to make demographic projections. So we view these mechanistic niche models as an integrative tool, and we are integrating other sources of information. So one of the major themes of our research is to do historic resurveys involving field surveys and experiments to try to figure out how organisms are responding in multifaceted ways to climate change. And we couple that by visiting museums and quantifying phenotypic shifts over time. And we also do a lot of work in more controlled environments, looking at physiology in the lab. So we work in two primary resurvey systems, both focused at high altitudes in the mountains of Colorado in the United States. So on the left, today I'm going to be focusing on our work on Coleus butterflies. And I also want to mention on the right that we have been and are increasingly working on a resurvey project for Colorado grasshoppers in collaboration with Cesar Nufio and other colleagues. And here we're starting to get more into the physiological and genomic mechanisms of climate change responses. So I'd be happy to talk about that with anyone interested. Okay, so for our case study on Coleus butterflies, the link between the environment and the organism that we're really focusing on is this phenotype of wing coloration. So these butterflies are closed-wing baskers. They orient relative to the sun in order to absorb solar radiation and heat up, or avoid solar radiation. And they have these light pteridine pigmented scales or these darker melanin pigmented scales on the underside of their wings. How many black scales they have on the underside of their wings is a really strong determinant of how much solar radiation they absorb and how much they heat up. And you can see this by looking at this elevational gradient in the extent of wing melanization. So if these butterflies are in these cool, high elevation environments, they tend to have dark wings that allow them to heat up. So hopefully fairly briefly, I am going to run through the modeling approach that we use to give us insight into how these Coleus butterflies are interacting with their environment. So here we're largely looking at elevation gradients, where we're looking at climate, and then we use microclimate models to translate this into the microclimate that these butterflies are experiencing on their host plant and in their environment. We can then use lab estimates of development time as a function of temperature to estimate phenology. So that tells us what time period of microclimate these butterflies are likely to experience across their life stage, and we can then estimate their pupal temperature. We then bring in wing absorptivity. So we start out with how wing absorptivity has historically varied as a function of elevation. We can couple that absorptivity information into our biophysical model to predict the body temperature that adults experience. And this phenotype of wing coloration is a really interesting phenotype to look at because it mediates responses to the environment at both very acute timescales and much longer timescales. So on the top there, you see a plot of the percentage of butterflies that are able to fly as a function of temperature. So you see they have a pretty narrow flight window. Their flight is very thermally constrained, and these butterflies need to lay eggs singly on host plants. So if we can estimate how long they can fly, that actually gives us quite good estimates of how many eggs they can lay in the field and thus their fecundity. If you look at the bottom there, you will see that egg viability drops off very sharply at these warm, acute, extreme temperatures in our environment. So we can integrate these longer term and shorter term responses to understand fecundity and also survival in these environments, and that gives us fitness estimates. Another interesting thing about this phenotype is that it's plastic. So if the pupa experience warmer temperatures during development, they have a lower value of absorptivity, as you can see, hopefully in that reaction norm on the lower left. And what we do to account for plasticity in our models, we describe the phenotypic trait both as the level of absorptivity at a fixed temperature of 20 degrees Celsius, and then we also describe the slope of that reaction norm relationship. So in this model, then, we can use a simple quantitative genetic model to look at these evolutionary feedbacks. Once we have optimal values for absorptivity from our models, we can create a directional selection gradient. We can use empirical estimates of heritability to project a response to selection. So hopefully the basics of our approach make sense to everybody. And today we're going to be focusing on alpine and subalpine coleus species. So at high elevation, there's coleus medii that has one generation. At lower elevation, there's coleus erythrale that has at least 2 generations per year. And the number of generations becomes pretty interesting, as we'll see, hopefully. So let's start out by looking at that high elevation coleus medii, and we can think about the fitness implications of wing coloration. So on the top, we have our projected fitness curve as a function of wing absorptivity for 1980, the start of our study period. And for those 2 sites, there's a subalpine site in red and an alpine site in blue. The empirical values during that time period are indicated with the data points and the confidence intervals. So you can see our models actually predicting pretty well what would be optimal in that environment. It's actually matching up with what is observed in nature. The other thing to note is on the bottom that we have seen quite a bit of climate warming over recent decades, but in these very variable montane environments, there is a lot of variability these butterflies are experiencing. And that was really the take-home message of this initial modeling. Interannual variability in temperature can really influence selection, causing selection to bounce all around the place. If you look at those fitness curves as a response to absorptivity for a subalpine site on the left and alpine site on the right, somewhat interestingly, you'll see the predominant trend through time as we go from early years in blue to later years in red is that fitness is increasing. These are really cold, thermally limited environments. So overall, it does look like climate change might increase fitness in these systems. But if we look at the left lower elevation site, we start to see the optimal phenotype shifting to the left to avoid overheating. And if we include those fitness curves in our quantitative genetic model, you can see the directional selection gradient on the bottom. So at our high alpine site in the right, we are again predicting a lot of interannual variation in selection that is really going to slow down evolution, but we are predicting consistent evolution of wing darkening to capitalize on this warming, basically extending the time period and the time period through the day and through the season that these butterflies can be active. At that lower elevation site, we do see as climate warming proceeds in our model, we are projecting a shift towards selection for wing lightening. And let's shift to looking at very similar dynamics for the lower elevation species, Coleus aurifili, that has 2 to 3 generations per year. So here we are looking at reaction norms, absorptivity as a function of temperature for 3 sites. So the highest elevation site is on the top, and we move through years from blue to red, being more recent. And without plasticity, you will see that our model is not predicting a lot of evolution. At high elevation, again, we're having selection fluctuating about 0, that's slowing it down, selection. And at low elevation on the bottom, we are seeing, interestingly, seasonal variation in selection among generations. So we originally thought these high elevation, really variable environments would have the most variation in temperatures these butterflies were experiencing, but we slowly came to appreciate that the really long season at low elevation can cause these butterflies to really experience a lot more variation in selection. So we can then look to see what happens if we incorporate plasticity in our model and the evolution of plasticity. And our models are suggesting that plasticity facilitates evolution, particularly at these low elevation sites, again, where there's a lot of variation in selection across generations. So you'll see this basically tipping up of plasticity projected in our model, and you will also see a lowering in the mean value of this absorptivity trait. So at high elevation, our models are projecting a mean decline with a slight increase in plasticity. And at low elevation, our models are projecting little change in the mean, but increased plasticity. And so those were looking over recent decades. And we are also projecting our models to look at biogeographic scales across Colorado and looking at future environments. So we are doing projections from 1950 to 2100 using a mid-range warming scenario, and we're looking at 1/8 degree grid cells. And so here in these plots, we're looking at the value of fitness components as a function of elevation. Every dot there is a grid cell. And we also look at 2 wing scenarios. So on the left, we have lighter wings. On the right, we have darker wings. And we're comparing here 1999 in purple to 2099 in yellow. And what you will see is that in the models for both flight activity or for flight activity on the top, we are projecting that with warming, there will be a decline in flight time available at low elevation, but an increase at high elevation with warming. And you can also see with darker wings, we have a shift towards basically shifting the optimal phenotype, or shifting the highest value of flight activity shifts to lesser or higher elevation on with darker wings there. And if we're looking at survival, egg viability on the bottom, we are projecting that climate change will reduce egg viability at low elevations. And we can also map this out onto the landscape of Colorado. And I'm not going to go through these plots in detail, but in general, we are projecting that actually this initial selection for wing darkening in the models might ultimately lead to darker wings despite climate change. But because we are projecting these evolutionary lags, this shift from selection for darker wings to selection for lighter wings, we might actually see evolution decreasing fitness in this system. So the usual way that people think about evolution and climate change is the questioning whether organisms will be able to evolve fast enough in response to climate change. So in this system, we're suggesting there might be a different dynamic where these evolutionary lags can decrease fitness. But again, plasticity really plays a facilitation role in this system by reducing the lags in selection time. Okay, so we've made a lot of predictions. Let's see if they hold up in nature. So this was work by Heidi McLean, who went around to a bunch of different museums. These phenotypes preserve very well in museum specimens. So we were able to look at specimens in 3 different regions over about 100 years. The higher elevation region that we tend to work in is on the left here. So interestingly, we do see this elevational shift in changes in wing absorptivity over time. So we see increases in wing absorptivity to higher elevation. Again, we think to capitalize on warming in these very flight limited environments, but we do see evidence of decreases in absorptivity at low elevation, where thermal stress is likely more of an issue for these butterflies. In work by Jessica Higgins, she was able to repeat historic lab experiments on plasticity. And we actually found no evidence for the evolution of plasticity on that top panel there, but we did look at gene flow, and it does look like high levels of gene flow might be preventing the evolution of plasticity in these systems. It's also the case that our models probably do not appropriately account for the genetic correlations and constraints in the evolution of plasticity. There is a little bit of evidence that there might be stronger plasticity at other elevation sites, but we don't have the historic data to test that. So to summarize this case study, hopefully really showing you the role of biology in mediating responses to climate change, our main message is that there's likely to be elevational variation in the interplay of plasticity and evolution. In this system, fluctuations in selection or slowing evolution, plasticity can facilitate evolution by reducing these fluctuations, particularly at lower elevations. And we are projecting selection for increased plasticity, but we don't see that empirically, so there are likely to be evolutionary constraints. We see this counterintuitive selection for heat retention at higher elevations. And really interesting to us, we are seeing a switch in the direction of selection with warming and this possibility that evolutionary lags might pose risk in response to climate change. So let's briefly shift over into looking at some of our tools. So this is called the TrenchR project, Tools for Translating Environmental Change into Organismal Responses. And a lot of these tools I'll be talking about have long lingered in textbooks for decades, and we are really trying to make them open, reproducible, better used, and we really would like this to be a community effort. So we would encourage you to help us out and get involved, and you can follow all this work on GitHub. So in terms of components, one component is TrenchR, which is an R package for transparent microclimate and biophysical modeling. So again, microclimate on the left here is scaling climate conditions into what organisms experience by the ground, wherever they occupy. On the right, we have biophysical models which allow you to conduct energy budgets to predict the body temperatures of organisms in their environment. And we do develop these tools in collaboration with Mike Carney and colleagues at NicheMapper, and they're quite complementary packages, but our version is a more transparent and more open version of those same tools. We also have a variety of visualizations to allow users to examine patterns of thermal stress, really look at how organisms are interacting with their environment. And for students, we have a series of educational modules that provide interactive data visualizations to look at how organisms have been responding to climate change. And we have a variety of other features there, such as profiling diverse scientists. And we are covering a variety of biological levels of organization and biological responses. We also have a series of more advanced educational modules for researchers to get introduced to this area. And quite recently, we developed a thermal image repository. Increasingly, thermal images are becoming more accessible with, for example, cheap thermal cameras that you can attach to iPhones. So we are really hoping that thermal images will be a great tool for researchers as well as the public to really understand how organisms are experiencing their environment and experiencing climate change. And we are also trying to disseminate our images on Instagram. So I would very much encourage you to follow us and contribute any images you would be interested in having us disseminate. So in summary, through this case study and then through discussing some of the tools we have available to enable this type of biology, hopefully I've encouraged you to think beyond air temperature to understand how organisms experience climate change. And again, I would encourage you to join us and contribute to this TrenchR project, your expertise and your resources such as thermal images. So thanks to our funders, particularly the National Science Foundation, and thanks to all the people that have participated in this work. And thank you for your interest and attention.
Well, thank you very much, Lauren, for that absolutely fascinating talk.
Sure.
Really interesting, and some fabulous images there at the end as well then. Just a reminder from the BES there in the chat, don't forget to add any questions that you have to the Q&A box. I'm going to start, if that's okay, completely selfishly with the question that I have. What do you see as the main barrier for getting people to include this kind of microclimate and thermal ecology in predictions of the impacts of climate change?
Yeah, it's not easy is the short answer. And you could get really bogged down with incredible spatial and temporal detail, trying to really characterize how organisms are interacting with their environment. So in general, we've been trying to overcome that by really thinking about the biology of organisms and what do we really need to include in the models to get the answers right, and what can we ignore. And I will say this microclimate relevant data has been really hard to come by in the not too distant future, but just over the last couple of years, there are a lot of really amazing tools that are coming out to much more easily account for how microclimate is influencing organisms. Yeah, and sometimes it's a challenge to figure out whether to rely on empirical data or do the kind of microclimate and biophysical models that we're talking about, but really it depends on the biology of the organism, and we do increasingly have a lot of different ways to reasonably account for the biology of responses to climate change.
That's great to hear, a hopeful response there. Okay, so Isabel Alonso has asked a question. Apart from climate change, many species will be confronted with reduced habitat and food source availability, so phenotypic changes may not be enough. Have you looked at interactions with, for example, changed food plant distribution or availability? So adaptation to temperature might be bad to reach food.
Great, yeah, that is a great issue. And I should have given the disclaimer that for the most part, we have been picking case studies to look at where we're really confident that temperature is the primary driver. For example, our butterflies and grasshoppers that we look at have pretty general and pretty widespread host plants and not a lot of strong interactions. So we've purposefully picked cases where we can really focus on the thermal response. But you're exactly right that a lot of these issues really are coming to play, particularly with the interaction. So we're increasingly looking at that. So for example, with our grasshopper system along an elevation gradient, we're increasingly looking at host plant availability and nutrition and how the thermal environment is going to affect the grasshopper's ability to digest those host plants and take in that nutrition. So yeah, that is certainly a future direction. And yeah, I would hope to do things like the Trench project, interact with people that are interested in thinking beyond just these thermal responses to climate change.
Thank you. And presumably the climate change could also affect the nutrition value of that food as well. So
yeah, I mean, that's the lesson of all of this, that it gets pretty complicated. So again, just thinking about the biology and trying to hone in on what really matters is usually the approach we try to take.
Okay, I'm going to come on to a question now from Wilco Verbuck, which is the talk emphasized the importance of temperature as experienced by organisms, but how about moisture and risk of desiccation, which is likely to interact with temperature?
Yep, that's the other main issue as a disclaimer of our talk. Yeah, we just know a lot more about the thermal response of these organisms and, for example, how this phenotype such as wing coloration can really mediate the thermal response. We know more than we do about precipitation. So that is why me in the field in general has been focusing on temperature, but yeah, it's a great point that thinking more about moisture availability and desiccation resistance is really important. Again, we picked systems where we could focus on temperature, but there is a lot of really interesting work coming out looking in particular at the interaction of thermal and hydric constraints and finding that there's some really interesting interplay there. So I very much agree that's an important thing to consider. We have increasingly been putting water balance models into our TrenchR resources, and we hope to keep on doing that. So anyone that's interested in that, we would greatly appreciate your help.
Brilliant. It sounds like that's the future. Okay, so coming back to kind of the start of your talk now, there's a question from Lorenzo Maria Iosia, I hope I said that correctly, which says, regarding trait-based predictions, do you think it might be eventually possible to identify good predictor traits?
I hope so. I think one of the main challenges is that we've just been using linear approaches and largely just single traits at a time. So a project in my lab, we've just recently been looking at nonlinear strategies and things like machine learning approaches that can really account for interactions among traits to try to improve predictions. And it is hopeful, I will say, it's not working as well as, like, we are getting better predictive ability, but not vastly greater predictive ability. So I do really think we need more case studies of how traits are mediating responses to climate change before we can really improve our model predictions. But yeah, certainly things like looking at thresholds, thinking about how traits are likely to interact. Hopefully we will be able to improve trait-based predictions. You know, we already can do a decent job, these trait-based predictions are also often used in things like conservation settings to assess species vulnerability. And we can get some information, but it is worrying to me just how poor a predictor sometimes our traits are. And I really think we do need to work more to improve traits as predictors. And it's a big task, but I'm also somewhat hopeful about our ability to do that.
Hope is always important. There's a follow-up question from Lorenzo here, which is, do you think a better understanding of intraspecific variability might cover an important role in predicting species responses to climate?
Yeah, that is a great point as well. And a lot of our own work has focused on characterizing interspecific variability. And I think, yeah, that gives us a ton of insight into how organisms are responding to different environments when they have other aspects of their biology fixed. And in particular, with more trait databases and everything, it's been really great that people are starting to think more about incorporating intraspecific variability. And I think, yeah, that's another great area of growth in this research area.
That's brilliant. Thank you, Lauren. One final question then from me, if it's a more technical question, if people are wanting to submit thermal images to your repository, are there any kind of standards that those thermal images need to meet, or is everything welcome?
For dissemination, we would appreciate any images you have. For our repository, actually, one of the main initial goals of it was to make FLIR thermal images, probably the most common image out there, make them so basically do a conversion so they can be used readily without specialty software and just used for analysis in something like ImageJ. So our repository is only taking FLIR IR images right now. And we do have some standards there, but if you guys have any thermal images that you want to disseminate, and ultimately we're also hoping to use the images to advance research and looking at how organisms are interacting with their environment. Yeah, I'm happy to talk to anyone that has thermal images. And I actually know the UK is a hotbed for great thermal images, and many of the ones that I know of we do not have yet. So please get in touch.
And of course, we have an international audience here as well. So
sure,
hopefully you'll find some winging their way in from around the world to you. Well, that's the end, I think, of our Q&A session now. So just to say thank you very much again for a really interesting talk. Lauren has very kindly agreed to answer any further questions on Twitter. So her Twitter handle and a relevant hashtag will be posted in the chat. So thank you once again to Lauren and to everybody for joining the talk today. Right, where are we? The BES will share some closing slides in a moment. Here we go, that's it. So next week at about the same time, we'll have Martin Nunez from the National University of Comahue, who will talk about why ecology must be a global endeavour. If you're watching this recording on YouTube, but would like to join future talks live, make sure you register for Ecology Live, which you can get to from the events page of the BES website. If you've enjoyed today's talk, you might be interested in our upcoming workshop, which is organised by the Climate Change Ecology SIG, on communicating climate change ecology and nature-based solutions to climate change. So this is a half-day workshop on Wednesday the 21st of April from 10 until 3:30 with a 1-hour lunch break. Don't worry, we won't make you work for that whole time. The registration deadline is next Tuesday at 5 o'clock. It's just been extended for the purposes of today. And the link to that has been posted in the chat by the BES. So if you're interested, do go and check that out. Finally, I'll leave you with details of an offer from today's sponsors, Oxford University Press. There's a discount code on this slide, and it will be posted in the chat as well, which you can use to receive 30% off their Ecology Live reading list, which includes today's pick for Lauren's talk, which is Southward's Ecological Methods, 5th edition. That's it from us. See you again next Thursday, and thank you again for joining us.